Ladies and gentlemen, welcome to the Lecture 8 in the Selected Topics of Biopsychology course in the SAS. That is our last topic in this class, and I hope you will have opportunity to understand the basics of anxiety disorders, which is our topic today, anxiety disorders. Let's have a look. So now you see in this street, in a sunny day, on a Sunday, or maybe, I don't know, but obviously it is summer or spring, and these people are walking. You see there is this couple here, there is another couple, there is this biker, there is this dog, there is this lady, and assume that something, do you think this lady could be scared of this dog? Yes? Anna, do you think this lady could be scared of, does she look like she's scared of the dog? Not. But the dog is pretty relaxed. But what if suddenly the behavior of the dog is changing and then the dog has become very aggressive? What would you expect the lady to feel like or to behave like? Scared, nervous, yes. Okay, so she would have fear, right? Of course, it also depends on the experience of the people with the dogs, with previous experiences. So depending on the people's previous experiences with events, there may be some different consequences. So that is also something we should understand when people is scared of what, for which reason. But in general, we have this emotion called fear, okay? And fear is important, and the learning of fear is important because it promotes defense against threats and reduces the need to relearn about danger. So it is important for our survival, because we just, remember and then we take precautions. And it is important at the same time to readjust our fear learning and fear behavior when the conditions change. So our brain has to somehow respond in a way that requires a kind of adjustment depending on the external conditions, that we also should we can learn to get scared of something, but when there is an adaptation, or when it is understood that there is no real threat, then maybe we should also unlearn not to get scared. Okay, so there is a matter of flexibility. That it is very important, but it should be also flexible enough to adjust. So if there is a problem in this flexibility, perhaps that's the problem that could contribute to anxiety disorders. All right? So when we talk about the anxiety disorders, let's define some basic terms and concepts. The fear, a negative emotional state triggered by the presence of a stimulus that has the potential to cause the harm. Anxiety, on the other hand, is a negative emotional state in which the threat is not present but anticipated. Okay, so anxiety is the anticipation of the threat. An anxiety disorder, these are the normal states that we all experience during our life. On the other hand, an anxiety disorder is a disease which requires treatment when it arises in the absence of any threat or disproportionate relation to a threat. So, okay, there is a threat, but the reaction is too much in comparison to the degree of the threat, and keeps the affected individual from leading a normal life. So fear and anxiety are normal states of human beings, and the anxiety disorder can become a disorder only if these conditions only in the presence of these conditions. And how do we diagnose anxiety disorders? Not we, but the medical doctors, the psychiatrists diagnose anxiety disorders based on the current textbooks, such as Diagnostics and Statistical Manual of Mental Disorders, as you know, or International Classification of Diseases. These are the two text guidelines that actually describe the criteria for the classification of mental disorders, including anxiety disorders. What I have told about this to you before, that this is very reliable clinically, but not validated biologically, because we do not have biomarkers. Okay, what is a biomarker? As you may remember, let me repeat, it's a biological characteristic that is objectively measured and evaluated as an indicator of normal biological or pathological process. It can be also used as a prediction for the treatment response of the patient. So there are different kinds of actually biomarkers. One type is diagnostic biomarkers, while there are other predictive and treatment response biomarkers. Unfortunately, we do not have such biomarkers available for the psychiatric disorders. So until we have the good biomarkers, which are objective, which will help and revolutionize the way how we treat mental disorders. Until that, we have to rely on these clinically very valuable guidelines. And according to these guidelines here in this figure, you can see how many different anxiety disorders already we have. So when we talk about the anxiety disorder, so you have to ask which kind of anxiety disorder a person has. As you can see here, separation anxiety disorder, selective mutism, substance-induced or medication-induced anxiety, specific phobias, other specified and unspecified anxiety disorder, social anxiety disorder, anxiety disorder due to other medical conditions, panic disorder, agoraphobia, generalized anxiety disorder, trauma and stress-related disorders, obsessive-compulsive and other related disorders. As you can see here, the dashed arrows show that actually these two disorders are currently classified under a different category in the latest version of the classification criteria of these diagnostic textbooks. That's why they are shown with the dashed lines. However, most of the time, since this is a recent change, and we are talking the accumulating literature, when we talk about anxiety disorders in general, we talk about all of these disorders. That's why it is, I think in this figure, they also show these ones with, despite with the arrows, but still they use this one. So when we talk about anxiety disorder, you have to think of this spectrum of these different manifestations of anxieties, anxious states. And what are those states? What are those manifestations? If you look at the generalized anxiety disorder, that is a case in which the person has continuous worry, tension, fear about everyday events, and mostly maybe for the near future, okay? And there is panic disorder, in which the person has repeated and unexpected anxiety attacks. There is panic disorder with agoraphobia, in which this condition is actually coupled with the agoraphobia, where the people experience fear of different conditions, such as leaving home, such as entering shops, such as going to public places. There are also social phobia, which is like a situation in which the people, having problems with socializing with other people. They avoid of social situations. And there are other specific phobia, for example, avoidance and fear of objects or situations, for example, for certain animals, for certain natural events, blood injections. And selective mutism is another case for anxiety disorder in which the individual can have some language competence in some situations while the individual does not have any language ability to communicate in some other situations. So sometimes he and she can talk, sometimes cannot talk at all. That is called selective mutism, and that is also common in the childhood. And then there is separation anxiety, in which there is an unrealistic and persistent worry about adverse events that might befall the patient's most significant others, or about other potential loss of same. So the people are worried about something happen to their to the loved ones. They will get away from them. They will leave them, although they are not leaving them. So nobody is leaving them. Have you ever experienced such a thing? Think of it. Of course, you may think of such things, but to make it like a real disorder, that is something here we just explained, but you see, unrealistic is important, and it becomes also so intense that it interferes with your life. So it interferes with your life in such a way that you just have this kind of worry about the loss of the loved ones. For example, when I was a child, I was maybe 7 years old. I remember some instances that at night, when everyone sleeps, I was worried that my parents, that's true. Now I am thinking, should I have some early signs of anxiety disorder? Well, actually, I don't think I have, but maybe I had a certain sensitivity, because when I was a child, I was scared that my parents would leave me and they would just run away from the window with both, they will hold their hands with each other and just run away from the window at night and leaving me alone at home. And I was so somehow stuck with this idea that I was waking up at night, going to their room and checking if they are there sleeping. So I had this kind of thing, but it was not something very serious, and it didn't cause like a serious burden. But I can understand that in the childhood, this could be a very soft sign, preclinical sign, and by the time, depending on a lot of factors, which we are going to see today, it could increase. It could be, such a manifestation could be a very early sign of something or not. We don't know yet. But eventually, later, of course, when I was grown up, I had different, child development is different, of course, is unique. So you may have such anxieties. But when it becomes chronic, that's something different. But this separation anxiety, this unrealistic way of thinking that you will be losing your loved ones, can also happen in children. So, what is the epidemiology of anxiety disorders? So what is epidemiology? I use this term sometimes, and I want you to know this before the final exam, that the study of the distribution and determinants of diseases is forgotten here, in specified populations. So that is the study of epidemiology. So what are the data regarding the epidemiology of anxiety disorders? So we know that approximately 1 in every 4 individuals is actually suffering or has suffered from anxiety disorders. But that means, you know, all these subtypes, you see how the spectrum is so wide. And the lifetime prevalence estimate ranges from 4.8% in China to 31% in the United States. That shows that there is a variation in different populations concerning the prevalence of the disease. For example, if you look at the country-specific one-year prevalence rates, again, you see that there are differences between the countries. So in different populations, there is a different prevalence of the anxiety disorders. Something to ask, why? All right. And here, if you would like to see some more epidemiologic characteristics of the disease of anxiety disorders, you're going to see that you see here in this, there are 2 bars here. Here on the right, you're going to see the 12-month prevalence of the anxiety, different type of anxiety disorder. Here, the first one is the 12-month prevalence of any anxiety disorder, which is about 14% or something. All right? And then, so any kind of anxiety disorder prevalence for a year. And then the second one, among the all types of anxiety disorders, we have the specific phobias that are in the second most prevalent one, and so on. Another important thing that there is a sex-specific differences in the prevalence of the anxiety disorders, meaning that women are more affected by this disorder compared to men. If you look at here, for example, in all disorders, this is the female-to-male ratio. Look at the female-to-male ratio more than 1. So everything more than 1 meaning that there are more female affected by this disorder. Is there anyone more than less among all these conditions? No. More than 1, less than 1? No. So it is more common among females compared to males. And the burden. What is the burden of anxiety disorders? As I already described, there are certain measures to quantify or to identify the degree of burden of a certain disease. And that are, for example, in this case, that is the disability-adjusted life years, DALY, which is a measure for disease burden. And the 1 DALY represents the loss of the equivalent of 1 year of the full health as a result of that disease. So that's a way to identify the burden of the disease for the individual and society. And if you look at this here, according to years with disability, you see the mental disorders actually ranked in the 6th place, and you see anxiety disorders, is ranked in the 6th place according to the years lived with disability measurements. And here you see actually days adjusted, disability adjusted life years measurement for 10,000 individuals for each diseases. Here, anxiety disorders are coming after depression. So they have quite high burden on the individual and society. So based on that, at least we can actually understand that females are more vulnerable to develop an anxiety disorder at any time during their life. Other factor is that actually children of individuals who have anxiety disorders, okay, have a 2-fold to 4-fold increased risk of developing anxiety disorders. That's another vulnerability factor. And of course, adverse childhood experiences are consistently associated with the adulthood anxiety disorder. But one problem with these kind of association studies is the fact that you get, so the people diagnosed with the anxiety disorder, and then you do a kind of survey in terms of their previous life experiences. Okay, and that is maybe not the best way to study this kind of associations. A better way of studying this is the longitudinal studies. In the longitudinal studies, there is a continuous or repeated measures to follow specific individuals over a period of time. Do you remember any longitudinal study which I described in our class? Arseni.
For example, study which was analyzing people with low
Exactly. Monoamine oxidase A gene variation and its association with the childhood abuse and its relation with the childhood abuse and the final outcome for the psychopathic traits. And that was a long-term longitudinal study. So that is also the case that was also followed for the studies related to anxiety disorders, which could give a little better hint about their pathophysiology and we actually understand that based on these longitudinal studies that anxiety disorders are actually usually start with childhood. Okay, and then sometimes the symptoms are not recognized, but they have some, in the preclinical case, there are some preclinical symptoms, and then later it can develop into a disease. Also, some anxiety disorders also are related to the other kind of disorders, other kind of mental disorders. So there is a high risk of developing other kind of mental disorders if you have anxiety disorder. As you look at here, and that situation is sequential comorbidity. So that means that the anxiety disorder is a predictor of another future mental disorder in the future if it is not treated. So here is the severity stage, and this is the actually age. And during the age, there are genetics, neurobiological, and environmental factors that could make a vulnerable situation for the individual to develop a certain case of anxiety disorder. And that early childhood actually symptoms that could appear until the age of 10. All right? And then in the presence of other factors, as the age increases, there is, high risk of actually already in the adulthood to develop an anxiety disorder together with another mental disorder. So, especially there is a correlation between the anxiety disorders and depressive disorders and substance disorders, but that is also something I put it with a dash line because, as you can know, this is something that we cannot really 100% claim. Why? As we always learn in this class that the pathophysiology of mental disorders are so complex. There are so many different factors. It is very hard to really make a very direct conclusion easily. That's why we are mostly in the stage of hypothesis. So there is a correlation between smoking, alcohol abuse, and anxiety disorders as well. And the question here is it something that anxiety disorders are causing the actually smoking and alcohol abuse because people, they are looking for some kind of relief in order to, maybe is it that, or is it the other way around, that there are certain systems in the brain are not functioning very well, certain circuitry is not functioning very well, and that also makes people vulnerable to alcohol abuse, for example. So the cause and effect relationship is not well known, but there is an association. And here in this slide, what is very important for you to know that these diseases are actually anxiety disorders, have a certain pattern to develop progressively. Okay, and there are some symptoms that you can see. For example, there are some subthreshold anxiety symptoms, which is the trajectory 1 here, as you can see here during the until here, for example, the situation in which there is this subthreshold anxiety symptoms during the childhood, the individual, there is this, so subthreshold, like not easy to detect and evaluate. And then in the threshold anxiety disorders, which is the trajectory 2, where you have here, and then there is this trajectory 3 in which there is comorbidities and secondary complications can be observed. So here the question is to detect the disease already in the preclinical stage and to take the precautions for disease to progress into a pathological state. So that is the actually aim of the preventative medicine. And that is the reason why we need to understand what are the etiology, what are the neural circuitry involved in this kind of diseases, that we can predict it before it happens, and we can perhaps find a way to fix the associated neural circuitry with preventative medical treatments or therapies or cognitive behavioral treatments of the individuals who are at risk before they develop the diseases. When they have the diseases, it's too late. In the modern medicine, the purpose is to stop the diseases before they happen. That is the goal. So in summary, today we know that most common type of mental illnesses is also anxiety disorders. All right, and we also know that approximately almost more than 60 million people are suffering from the anxiety disorders. And we also understood that any anxiety disorders often begin during the childhood with unrecognized symptoms. And it is more common in women, and anxiety disorders elevate the risk of developing other mental disorders. So now let's talk about the background. What is it? What happens? How does it actually, why does it occur? We understand that there is this fear and the anxiety, which are natural states, but there is somehow abnormal anxiety. That means that the system associated with that fear, learning and anxiety is dysregulated for some reasons. So then people would like to understand what are the genetic factors associated with these conditions. Why we say like that? Because we know that majority of the mental disorders occur due to the genetic variation and its interaction with the environment. So there is this gene-environment interaction, but you know, some people, no matter what the environmental adversity is, they will not get anxious. They will not fall into the disease of anxiety disorder because their genetics is not for that. Some people, if they are lucky, if the environmental adversity is okay, is not so bad, then they will also not develop an anxiety disorder. But that is because they are maybe grown up in a better environment without too much stress, some adversity during the childhood. So you see, we cannot determine our genetics. We cannot, during the childhood maybe especially, we cannot really control our environment. So there is this gene-environment interactions, but we can, if we can understand how these gene-environment interactions lead to the development of certain disease states, then we can actually determine the individuals who are at high risk already from the childhood, and we can take some precautions before the diseases are being developed. So these are the actually for the genetic factors, again the genetic variation effect of the genetic factors is clear because there is this degree of heritability which is in between the 30 to 40%, all right, and that is actually not something to ignore. That's quite high. And that is already clear with the previous epidemiological data in which I described that, you know, you remember maybe when I said that individuals whose parents have an anxiety disorder, there is a high risk of developing anxiety disorder. So that already shows that there is a genetic component, and that is like in this range. And there seems to be that there is a genetic overlap between the different anxiety disorders. You see all these different kinds of anxiety disorders, their symptoms are different, that's why they are classified differently, but there is a genetic overlap between them. So that means that certain genetic associations are common in all these kind of diseases. At least for the moment we think like that, but tomorrow maybe there will be some new data and maybe this will be rejected. We don't know. I think so far you already have understood that how actually genetic analysis of the mental disorders is changing and shifting, And maybe everything what we have learned so far will turn into a different situation, especially as a result of the new analysis methodologies such as genome-wide association studies, which I described when I talked in the course about the depression. But there is somehow this kind of overlap, but also there is a genetic distinction as well. And that distinction is especially clear between the, fear versus disorders characterized by negative affect, like generalized anxiety disorder and phobia. They are a little different. And then there seems to be a kind of maybe a genetic distinction between them. But we still do not know. They are like ghosts. What are those? What is the genetic architecture behind of the whole spectrum of these diseases? We don't know. So just a brief summary from the previous class. Also just remember that how actually people find out the genetic factors involved in the etiology of anxiety disorders or any kind of complex disorders. Typically, it actually was done by linkage studies and association studies. Especially in the association studies, in the classical way, there was this candidate gene association studies. And there were so many candidate gene association studies have been conducted in the whole different diseases of psychiatric conditions. But unfortunately, they were not replicated. Replication is very important. That's another important thing I want you to learn in this class. We do not just rely on one scientific publication. It has to be replicated by independent groups with independent study designs. And we didn't have such a good replication rates in those analyses that were achieved by candidate gene association studies. And then, of course, with the advent of new techniques and statistical approaches and sequencing methodologies, there are more robust methods to analyze the genetic variations in a large-scale fashion in which you work with 10,000 people with millions of different genetic variations at once, and that gives you maybe to catch some kind of correlation. And that is the genome-wide association studies. And we are at the age of the genome-wide association studies. Every time their prevalence, these kind of studies are increasing in the literature with better designs. And we are almost trying to actually catch the genetic associations involved in these disorders. And for example, for depression, genome-wide association studies, as we already discussed in the previous class, those previously famous candidate genes were not actually found as the most significantly associated genetic variations. So completely something which is not very much associated or thought to be related with depression turned into something new that has the highest significant association with depression. So that is very interesting. And none of these actually new candidate genes that are found as a result of genome-wide association studies are the targets of, for example, antidepressants. I mean, their gene products are not directly targeted by the antidepressants, such as monoaminergic modulators. So that's very interesting. That's why, as we already discussed, that we are waiting a kind of a revolutionary turn point in the understanding of the mental disorders, and then that will be followed by their treatment in the future. So these are the terms that I just added for you to check later. Why do we know that there must be genes associated with anxiety disorders? Because of the heritability studies. Heritability studies show that this is not an environmental disease. There is a genetic component. Okay? So there is gene-environment interactions there, and the genetic component is pretty high, almost 50%, 40%. But what are those genetic factors? There are genetic factors, but what are they? That's the question. I can tell you different genes, always, that if you just Google genes, genetic factors, anxiety disorders, panic, whatever, you will find some genes that are probably found as a result of candidate gene association studies. But as I said, they are not replicated, well replicated. So these are all in the theoretical pool of different data, but are not potentially translated into the practice, into the medical practice, into the treatment of mental disorders, into the way that we can find and use them as biomarkers. No. For that reason, I can already list some examples that have been at least with some correlations, with some good correlations that have been found to be associated. But as I said, there is this replication problem. Monoamine oxidase A, the famous monoamine oxidase A, is everywhere. So monoamine oxidase A is related to antidepressant response. There are monoamine oxidase A inhibitors. Monoamine oxidase A genetic variation is related to psychopathic traits if the individual was exposed to adverse childhood abuse. So it is like you cannot just use and take one gene that can be correlated with one disease condition. And if you already understand how the brain structure, how the neural circuitries, how all these different neural cells and ensembles forming some, functional features, you can understand that is, to some extent, there might be some common mechanisms. That's why you see certain genes everywhere. But what are the ways that they get differentiated from each other, that there are different ways of diseases, disease manifestations? So pretty hard question. So that's why this is an example for you to show the genome-wide association studies, just briefly to remember from the previous class, but I will not spend so much time on it now. I want to actually talk about brain regions involved in the processing of fear and anxiety. So in order to understand that pathophysiology, we want to understand overactive or underactive regions that are, that are primed as a result of the childhood adversity. So certain regions are more active than it should be. Certain regions are less active than it should be. And that happens because of the experience. And then that does not get better. So that needs to be understood well. And another important thing is measurable aberrations in these systems might predict the later presence of a disorder. So because we need to know this in order to really predict specifically if a specific case of a mental disorder, especially in this case, anxiety disorder subtype, is going to develop. So then we can maybe fix that circuitry before it develops. So, but we have the problem of heterogeneity of anxiety disorder. So there is like all these different kinds of spectrums of anxiety disorder. There is also comorbidity that, a certain anxiety disorder can be associated with another comorbidly existing with another anxiety disorder. Plus, some individuals can have anxiety disorder plus other kind of mental disorders. So how actually resolve all these situations, such a heterogeneous situation? That is a problem. And then how actually do we have a common ground for anxiety disorders, or they are completely different? Okay, they are classified under the group of anxiety disorders, at least in the level of phenotype or symptoms. There is a common ground. But that common ground, is it reflected also in the biological substrate, that there is a common mechanism behind of that or not? That is the question. And that is a difficult question. But That's why, but still there may be some common ground, and that common ground has been found as a result of different kind of studies. These studies basically can be summarized as the studies that are dealing with the analysis of the processing of negative emotions using different tasks in humans. So, you know, for example, you take the people under the brain scanner and then you show them some different kind of stimulants that could trigger fear or associated feelings. And then you can actually measure the brain regions that are active, and you can compare that with the normal people. And you can also actually compare it with their genetics as well, and you can make an overall analysis. And that actually could give a kind of hint. And that was giving actually a kind of hint. Other kind of way of use of Pavlovian fear learning procedures in humans and rodents. So these kind of studies have led to the identification of a kind of a certain common ground to some extent. This is actually the way, but I wanted to show you heterogeneity. But so if we take just one type of anxiety disorder, let's say general anxiety disorder, and then certain features, features that are observed in the brain structures can be summarized in the slide. That certain gray matter volume is increased or decreased compared to normal patients, especially in the amygdala, for example, the gray matter volume is increased. But if you want to understand the common ground, then especially different studies, for example, non-human species based on lesion studies, or knockout or optogenetic animal models were used also to understand some basic circuitry involved in, for example, fear learning. Case studies that are also important in the whole different topics in neuroscience that we already discussed in the case of, for example, HM, as you remember. But here also there are some case studies unique that are important or critical for understanding of the anxiety disorders, which I will describe today. Electrophysiology and functional neuroimaging studies, for example. So human neuroimaging studies show especially the amygdala and its association with anxiety disorders. But I want to, especially being the neurobiologist, of course, I want to more speak something related to my background. So I want to show some example of knockout or optogenetic animal models and how they could actually help us understand mechanisms or circuitry involved in the pathophysiology of a specific anxiety state, anxious state. So optogenetic is a methodology in which you actually modify specific cells, very specific cells, billions of neurons that are found in different anatomical structures that we already covered in the previous classes, how these structures are so complex. And then there you go, one specific brain structure, and then you just, there in one specific brain structure, let's take amygdala, there are so different groups of neurons, nuclei, each are organized in a specific way, and then you choose only one subtype of cell there, and you modify that cell genetically, and that cell after that becomes sensitive to light. So optogenetics is a methodology to modify certain cells in the brain, at least for this course. You can also do it for other systems, but you modify genetically and specifically certain subset of cells that they are active or they can be sensitive to light. And then you can actually make the cells excited or inhibited with the light, depending on the way of manipulation. And by that way, actually, you can actually control the neural circuitry and associated neural region and maybe behavior. And that is an example of actually such a system here. This is the laser light that has the potential to actually specifically send a specific light with a specific nanometer in which the brain cells have become sensitive is exposed via this laser. But now now there are the wireless lasers as well, that you can actually put a detector that the organism can receive the light signal wirelessly. So there are already studies that are actually being done to address the circuitry. Amygdala circuitry, look at this sentence, that's very significant. Amygdala circuitry mediating reversible and bidirectional control of anxiety. So in the animal model, they found, so they modified a certain region in the amygdala with the certain groups of neurons are modified in the amygdala. And then they actually, what they did, they basically, when they activated or inactivated it, they found a situation in which the anxiety is induced or blocked bidirectional. So that is something very profound. So what is this? This is a slide from my latest publication, and I wanted to use it in this class. So these are actually different proteins. This is a cell membrane, and these are different proteins. And there is something common for all these proteins. These proteins are belonging to different organisms. For example, this is from algae. This is from bacteria. These proteins are called opsins. Okay, for example, in the retina, we also have light-sensitive proteins, right? Do we have light-sensitive proteins in the retina? Yes, we do. That's the way we see. These proteins that are found in the retina, they have the ability to convert the light signals into the electrochemical signals. That electrochemical signal transformation is just mediated by changing the depolarization or voltage potential of the membrane, as we already discussed. So how is it related with these opsins? So these opsins are something unique for these molecules. They are all called opsins because they have some certain common features. And the most important common feature is that they are sensitive to light, but specific wavelength of light. For example, this is sensitive a certain wavelength of the light, whereas this is another wavelength, and this one is sensitive to another wavelength of the light. What does it mean it is sensitive? Once this is exposed to specific wavelength of a light, which I will show you, the channel of this protein gets open. When it gets open, it will allow the passage of the ions to which it is permeable. Ladies, right? And its permeability is actually different depending on the type of the channel. This channel is actually sensitive to cations. This channel is sensitive to chloride, and this channel is sensitive to hydrogen, protons. And this one is sensitive, but the way in which direction the ions are going to flow through the channel depends on the type of the channel as well. For example, this one is a proton. This is proton. A hydrogen without one electron is hydrogen ion, which is also called a proton. This is a proton pump. A proton pump. When I say pump, that means it doesn't matter how much proton exists here. Okay? In this case, it is actually sending the proton outside, let's say. But even there are less proton in the inside compared to outside, since this is a pump that will actually send the proton against its concentration gradient. But in this kind of receptors or channels, the concentration gradient is very important. That the concentration of the ions and the charges of the ions are going to determine to which extent which ion is going to go to which side of the cell. So all these different properties will allow us to use them if we can actually put them into the specific neural cells. That will help us control the excitability of the neurons. For example, if I put this kind of protein, which is isolated from an algal species, and I can actually introduce that to the cells of the mice brain with genetic engineering techniques, and then I activate the channel with the specific light, the blue light. It is sensitive to blue light. And then once the blue light comes, there will be some kind of flux of the positively charged ions into the cell, which is found in the mouse brain. Or you can actually put a green light, because you already inserted by genetic engineering techniques this protein into the brain of the animal, or yellow light. And actually, I want to show this technique here. It is very elegant. It sounds sophisticated when I speak like that, but actually it's not so hard. It's not so difficult. We use viruses as gene delivery machines. Here, you actually put the gene. For example, this is the channelrhodopsin gene here in the virus DNA. And then you pack the virus. You can make the virus. Do you remember there was this discussion whether coronavirus was done in the lab? Do you remember? There was this theories. But actually it's pretty easy to do viruses in the lab. And there are different kinds of viruses, and some viruses are not very safe. Because, for example, antivirus here, then there are special precautions and systems in which you can actually work as long as you have the safety requirements in the laboratory. So you have the antivirus DNA, and then you introduce in that DNA the desired DNA that you want to put into the viral DNA. And then you pack the virus with some additional, these are the technical terms I don't want you to know. All I want you to know that you can put this gene encoding for this protein, gene encoding for this protein, or gene encoding for halorhodopsin here. Here, this is the channelrhodopsin gene, or any other gene. As long as you put this here, then this is a laboratory procedure. You produce the viruses that are having this gene inside, and the virus is going to be injected in the mice in a specific brain region. And you can think that, but how can you just so specifically allow these viruses expressing those proteins into specific cells? Because brain region injection is not so specific, right? You have such genetic modification techniques that you can allow to make this drive or expression to work only a selected subset of cells in a given brain area. So you can actually make an injection into the amygdala, and there the viruses are going to diffuse through the whole amygdala, and they are going to actually, infect all cells that are found in the amygdala. But the expression of these proteins is going to happen only in selected cell types, because you know how to adjust it. There are ways how to do it. And by that way, you have a transgenic mice which has in its specific brain cells that are having very tiny, because this will be expressed as a protein, that are having this protein in their neurons, and then you induce the light, and then you can control the neural circuitry in amygdala, and you can make the check whether the organism is changing its behavior as a result of activity, as a result of light-induced activity of those receptors. And why do we use those receptors? Because we want to change neurons' activity. And we change the neuron's activity physiologically by actually inducing the light, and the light gets this process changing, and the membrane potential of the neurons that are having these kind of receptors or channels are going to change. Okay, this is an example of optogenetics. And after that, you can also put the animal into different contexts and under the different learning paradigms and study the conditioning, fear learning. And whether if you overstimulate or stimulate that circuitry that you already manipulated by optogenetics, you can study and you can find out all associations. So another kind of studies are the case studies. In the case studies, which I will show you now, that one famous patient is the patient SM. So she was an American woman with a peculiar type of brain damage in which there was a complete bilateral amygdala destruction. So that is, you don't take and make a lesion, of course, but there are some specific cases in which the patient itself has a lesion in their body and in the brain, and as a result of a specific disease condition. And of course, that's an excellent source for scientific analysis. So there is this lesion in the amygdala. And what would you expect this lady when she does not have her amygdala here? If you look at this study, which was published in 2011, here there are the different sections of the brain of the patient. This is A, this is B. A represents the lesions in the amygdala when the study was done in 2011. B represents the same condition actually exists when the patient was actually, because the patient was followed for a long time. So before already she had this condition for a long time. She had this amygdala lesions already. And then when she was taken as a case study subject, then that was, her brain was scanned again. And they also did show here that these regions of the amygdala are, so the amygdala is not, I'm trying to say that there is a bilateral lesion of amygdala. No amygdala. So what do you expect this lady to behave like? They wanted to study, and they did a lot of studies on this patient. One example here, but I want to tell you the most significant experience of her later. But let's talk about some data. Here, for example, this is her feelings during a specific time period. And then what she has experienced as feelings have been recorded. So she has sadness in different, manifestations. She has a feeling of anger in different levels of manifestations. She has the feeling of surprise. She has this feelings of happiness in different manifestations. She has disgust, but she has no fear. No fear, no fear. Fearless lady. Also, they compared her, they showed actually this individual movie clips with different kind of characteristics that could induce sadness, anger, surprise, happiness, disgust in normal individuals, and they compare it with this patient. And they found that she has actually sadness, so this is the healthy controls, this is the patient. And they show the movie clips, and for example, movies that potentially induce sadness in the normal people also induce sadness in this patient. Movies that potentially induce anger in the normal people also induced anger in this patient. Movies that induce happiness in normal people also induced happiness in this patient, and so on. But fear, the movies that induce normal fear in normal people did not induce fear in the patient of SM. So that is very profound, but perhaps one of the most interesting experience in her daily life was the following. So she was walking at night in a park, during which she was attacked by a man who had a knife. And the next day, she went park again. She had no feelings of anxiety or fear. On the other hand, yes, this is a kind of a case study that makes us think that amygdala is a very central brain station that could be involved in the anxiety disorders. But we also know that not all anxiety disorders are dysregulated in not every case of anxiety disorder is associated with the dysregulation of amygdala. For example, here, these are the patients with lesions in the amygdala, but these patients have, for example, panic anxiety. So you see, it's pretty hard to conclude, but we can already say that probably panic anxiety can be different than other kinds of anxiety. So, but no matter what, I just wanted to give you an example of the knockout, optogenetic, not knockout. Knockout studies actually studied a specific gene that is not expressed in amygdala. They also found that those animals are fearless animals, in which the specific genes that are found in the amygdala, statmin gene encoding for the statmin, which is a specific protein, especially expressed in the amygdala, is knockout animal. And then they found that those animals are fearless. They don't fear like other wild-type animals. And we discussed about the case studies, and there are also neuroimaging studies. And as a result of that, we have one actually case, one maybe if you want to make a gross anatomical description of the dysregulated brain regions that are found in the anxiety disorder, perhaps we can say that these regions, as a result of all these different lines of evidences, let us understand that perhaps there is this amygdala that has a bidirectional interaction with especially the prefrontal cortex, anterior cingulate cortex, hippocampus, and insular cortex. And all these interactions are bidirectional. So that means like there is somehow this kind of different neural circuitries that can be somehow found as dysregulated in the manifestation of anxiety disorders. And yeah, somehow not to forget, when we talk about anxiety, probably amygdala now, after all. And just remember the amygdala, as you can remember that it has actually the basolateral nuclei from which it gets the sensory information, as I already described in the previous class. And then from there, this is the central nucleus that actually drives different signals that can be associated with the stress response, which is the, for example, hypothalamus pituitary adrenal axis here, hypothalamus pituitary adrenal axis, which actually releases the cortisol. And that's supposed to go to hippocampus, and there is this here, hippocampus is supposed to be stimulated, and then there is this feedback loop that should make this HPA activity shut down. But somehow this circuitry also is not maybe functioning in the people who are having different kinds of anxiety disorder. And the treatment of anxiety disorders. Do you know what are the treatments for anxiety disorders? This is famous. This is my topic because diazepam is a drug that is acting on the famous receptors, GABA receptors. These are the primary inhibitory receptors that are activated by GABA, and they actually dampen the nervous system, meaning that they actually decrease the activity of the neurons. They inhibit the neurons. So that is a little like, you know, if you think about inhibition of the neurons and if you think about the anxiety state in which actually your brain is overactive, it's interesting that there is somehow such a correlation between a physiological inhibition and psychological inhibition. That's something to be dampened. But not necessarily. When we think of the brain, we cannot really say that inhibition always is good and excitation, it means vice versa. To some extent, for some circumstances, yes, but we cannot make such a linear correlation between the physiological inhibition and excitation and psychological manifestation. Okay, so when I say excitation inhibition, I talk about really physiological depolarization of the neurons, but I'm not talking about the behavioral excitation. It is not easy to make such a correlation, but somehow it sounds like similar. So, but in this case, it is overlapping, right? So there is this anxiety disorder and anxiety disorders. Sometimes these are the second line, not the first line. The first line treatments of anxiety disorders are usually selective serotonin reuptake inhibitors, which are the drugs that are used also for the treatment of depression. So you see, again, the serotonergic system is again in the treatment of anxiety disorders as well. Probably because of the diffuse modulatory system. If you remember the diffuse modulatory system of the monoaminergic neurotransmission, they come from the brainstem and they travel through the whole, you know, cortex and cingulate cortex and everywhere, and you just change something there. You know, modify something without eyes are. So think about like a pipe system. There is something wrong in that pipe system and your eyes are closed and you are trying to, okay, now it's okay. It's like that for the moment, unfortunately, how it is treated. And you feel okay? Okay, keep going, keep taking. But there are a lot of side effects. There are problems of, for example, worsening the symptoms in the first days of the treatment when people are treated with these kind of disease medications. So we don't have really very, you know, excellent pharmacotherapy. Of course, there are also behavioral therapies that are very important, especially cognitive behavioral therapy should be very valuable, or combination of both. But since we are talking more with the circuitry here, I was more focusing on the chemicals. So, unfortunately, we don't have new exciting news for the development of the new pharmacotherapy. But I am still very optimistic about these diseases and any other diseases that are found in the psychiatric domain. I am quite optimistic. I think for this course, you already understood that science is really working hard with all these very sophisticated techniques, with sweat, try to find out the way how actually these things happening, why happening, and then try to block them before it happens, as a practice of preventative medicine, and try to find out really the biomarkers that are defining the specific disease cases and defining the diseases according to those biomarkers, and then trying to medicate those diseases in relation to those biomarkers will likely happen in the future. That's why I'm very optimistic, despite all complexity and all problems that we have with the burden, with the problems associated with the psychiatric disorder, including anxiety disorders. I am very optimistic, and I am sure you are also convinced when I just show you some little bit of data and sophistication of the works that have been done in this field. You should be also optimistic.